So today we're going to focus on mutations. There are no real new mutations based off of what you have seen before your freshman year. So again, it will be the application of these mutations. So a mutation in general is just going to be a change in our DNA sequence. And that mutation could be good, it could be bad, and we'll get into that in a little bit. But the basic definition of a mutation is just that there has been a change in the DNA sequence. And these changes could alter both the type and the amount of the protein produced. So they could change, and now you're producing basically a different protein, or you could produce more of a protein or less of a protein. They also, in turn, could change the phenotype. Remember, phenotype is dictated by the DNA, by the genotype. Phenotype is dictated by proteins that are made, because that's all DNA can do is make proteins. So all the physical characteristics that we have, all the things that make our body function, all the hormones that are produced, all the skin color, the hair, the height, the brain function, it's all controlled. When we say it's controlled by the DNA, it's really controlled by what proteins the DNA is producing. So if I have a mutation, if I change the DNA sequence, that could end up altering the final phenotype. Okay? The benefit to these, though, is that it's going to help cause genetic variation, which we like, right? Remember, that was the whole purpose of mitosis, was to help cause genetic variation. So the normal function of genes and their products gives me the normal function of the organism. So if I alter or change it and I get new phenotypes, that could be good, that could be bad. It could, a brand new phenotype is either going to be good or bad. It's not really going to be a silent situation. Okay, but these mutations are definitely a source for genetic variation. Let's look at an example of one. So let's look at cystic fibrosis as an example of a disorder that's caused by a mutation, right? Because if I have normal functioning genes and they're producing normal functioning products, then I'm going to have normal function of the organism. But if I have a mutation that's affecting my proteins that are being produced, then I may not have normal function of the organism. So let's look at cystic fibrosis example. So cystic fibrosis affects primarily whites of European descent. It hits about 1 in 2,500 births. So on average, 1 out of every 2,500 babies that are born will be diagnosed with cystic fibrosis. 1 in 25 whites is a carrier. So cystic fibrosis is a carrier disorder, so it's a recessive disorder. So 1 in 25 whites carry the disorder. The normal allele codes for a membrane protein, CFTR. Okay, and so the normal allele codes for this membrane protein that will transmit chloride across the cell membrane. So you can see, here's my CFTR channel in this first picture here. And you can see the chloride ions are able to transport across the membrane. You see they're leaving the cell, and they're going from the inside of the cell to the outside. So that's our normal functioning. So no mutation there in the DNA, then I get this normal functioning chloride ion channel. If that channel is defective, then it limits the transport of chloride. If you look in the second picture here, I have the defective or the mutant CFTR channel, and you'll see that it's closed. So if it's closed, chloride isn't moving across the cell membrane. Okay? And water, remember, water has a charge. Chloride is a negative ion, right? And water is polar. So water can follow that chloride ion, and it doesn't. So because the chloride ion isn't leaving. So if I have the mutation and I have the mutant CFTR channel that's closed, then I don't have as much chloride and water moving across the cell membrane. And the result is the mucus on the outside of the cell gets really thick because it's not being thinned out by the water. So it gets really thick, it gets really sticky. Okay, so we get this thicker, stickier mucus coat around the cells. And that will build up in certain organs. It'll build up in their pancreas. It'll build up in their lungs. It'll build up in the digestive tract. Okay? And the buildup of that mucus can cause bacterial infections, which can be obviously an issue for these patients. So without treatment, children usually die before the age of 5. However, with treatment, they can live up into their late 20s. More and more advances are being made, and there are patients that are making it into their early 40s. The issue here is, again, you can see in the airway in the top right here that they're having trouble, they'd have trouble breathing because of the thick layer of mucus that's there. It restricts the airflow. If they're getting these serious bacterial infections, in particular in their lungs, that's usually the result of death. And this all comes from a mutation in the DNA that produced a defective protein. So let's look at some different types of point mutations, different types of mutations in general, in particular point mutations. So these point mutations, these are a single base change. So these are gene mutations. These are mutations where one nucleotide is affected. So small little gene, not big chromosome mutations, but gene mutations where one single nitrogen base has been affected. So one of the most common thing here is going to be a base pair substitution. So I substitute one base for another base. So when I have this base pair mutation, I can have three basic things that can happen. I can have what's called a silent mutation. So this silent mutation right here, it has no effect. Okay, so if I look at my original on the top, I'm producing methionine, lysine, phenylalanine, and glycine, and then a stop codon. So with this base pair substitution, where I substituted a U instead of a C, I still have, it has no effect. And so since that will not affect the phenotype, that is considered a silent mutation. There is no amino acid change, so it's neutral. Right? These are also neutral mutations. So there's no amino acid change, and so therefore there's no effect on the phenotype. This is going to be due to that redundancy in the code. We talked about this on the last video, how we have we have multiple codons for one amino acid, and so that redundancy in the code allows for these mutations to happen and them not to cause a problem. Not to always be negative because of that repetitiveness of the genetic code. My next option is what's called a missense mutation. So a missense mutation changes the amino acid. I can see right here, I substituted an A for a G. I get serine instead of glycine. So it's just changing that one amino acid. This could be a good thing. It could be a bad thing. It could have no effect. So this could be what we call a positive mutation, it could be a negative mutation, or it could be a neutral mutation. Maybe it produces an enzyme that makes the reaction go faster, so it's a positive mutation. Maybe it produces a enzyme that doesn't work now because it changed the shape of it, so it'd be a negative mutation. Maybe it has no impact, so it's a neutral mutation. Our last thing is we could have what's called a nonsense mutation. So a nonsense mutation, you can see it here, I'm substituting the U instead of an A. A nonsense mutation changes that amino acid to a stop codon. This is always going to be a negative mutation because it's going to stop production of the polypeptide. You'll notice after the stop codon, I didn't start again. Okay, now I have phenylalanine, now I have glycine. Like I didn't do that. We don't keep going. It literally means stop. That's how the cell knows that the protein is over and we don't need to keep adding to it. So whenever I get a stop codon, it will do that. And so that's going to end up being a negative mutation. You're cutting these proteins short, so they are definitely not going to work like they should. You can also have point mutations here where you remove or delete one nitrogen base or add or insert one nitrogen base. And these mutations can be an issue. They could be neutral, they could be positive. They're very rarely neutral. They could be positive, they could be negative. Our issue here, what will happen here, is that when I delete or insert that base, I completely shift my groups of 3. So if I have, okay, so if I have my groups of 3 on my original DNA up here, or mRNA here, I have my codons here. If I add or I subtract one base from that, that will alter what's called my reading frame. My groups of 3 are my reading frame. How they are grouped into these threes, into these codons, that's my reading frame. But as soon as I add a whole nother, let's just add a second G here, right there at the beginning. So now my groups of 3 have shifted. So everything after where the mutation occurred is going to be different because I've shifted my reading frame. I've shifted my groups of 3. We say the same thing if we subtracted one. Okay? Instead of adding a G, let's subtract the G. So now, again, my groups of 3 are going to change. So now I get to these as my groups of 3. So I've shifted how I read it, how I group it. And so insertion, deletion mutations will do that. A point mutation that is a base pair substitution will not do that. But a substitution can give us these missense, nonsense mutations. So what we have here are some mutations that can happen on the entire chromosome. So these are bigger, larger chromosomal mutations. So I could have a deletion where an entire chunk of the chromosome is missing. Not one little nitrogen base or one nucleotide, but an entire chunk of chromosome missing. I can have a duplication. You can see here where I've duplicated it. Both of these are errors that could occur while DNA replication is happening. I could also get an inversion happening when I do crossing over. Take a piece of the chromosome out, reinsert it, but reinsert it upside down, as well as a translocation. A translocation is going to require 2 chromosomes for this to happen. A translocation is basically crossing over with non-homologous chromosomes. So I move one chromosome segment to another, an entirely different non-homologous chromosome. And that is a huge problem. Some things that can cause mutations would be errors that are happening during DNA replication. If you remember with DNA replication, DNA polymerase 3 is the one adding my new nucleotides onto that 3' end. And it adds 1,000 base pairs per second. That is super duper duper fast. And so it makes mistakes. Okay? It will occasionally not pair an A with a T or a C with a G. It will make mistakes on occasion. The enzyme will pair the wrong ones, and that can cause a mutation. DNA polymerase 1, which helps replace those RNA primers, will also try to help edit and fix the mistakes that were made while the DNA was replicated. The problem is it doesn't catch every single mistake. And so if it doesn't, there are certain mistakes that it could catch that it could miss that could be detrimental. So there are environmental or external factors that can also cause mutations. These things are called mutagens, right? So something that can cause a mutation is a mutagen. Things like UV radiation, so from the sun, reactive chemicals, radiation in general, like radiation from an X-ray, these can all cause mutations. The mutations, remember, again, they're not always bad though. So they are they're one of the number one sources of genetic variation. Okay? But they could be beneficial, detrimental, or neutral. If they're detrimental, they usually won't hang around because that particular organism will not reproduce and pass its DNA, pass its mutation on. Okay? If they're beneficial, they'll usually remain around for a long time. If they're neutral, you may not even know they were there. So mutations that can happen during mitosis and meiosis. That's a pretty relatively common place for them to happen because these chromosomes are separating. You're replicating DNA as well as then separating homologous chromosomes or sister chromatids. And so they could separate improperly, and we already talked about how they could replicate and miss some of the mistakes. A lot of times these particular mutations can result in a brand new phenotype. For example, a triploidy, so instead of having a pair of chromosomes, instead of having chromosome pairs, you end up having a triplet, like in Down syndrome, where you have trisomy 21. So the fetus is born with an extra set of chromosomes, which will cause a number of defects and sometimes can result in the death of the fetus. And the ones that do survive are usually sterile because they don't want that to continue. They don't want that to reproduce. Sometimes it's plants is called polyploidy, where the plants end up with these mutations, is not a bad thing. It can be very important to their evolution by basically adding or removing these particular chromosomes from their DNA set. A lot of plants that are cultivated agriculturally have experienced some sort of this polyploidy. Wheat, strawberries, bananas all have. And we'll talk, this is kind of a brand new thing, so we'll talk a little bit more about that in class. These changes in chromosome number, though, can result in very significant disorders. Like we said just a minute ago with trisomy 21 for Down syndrome, Turner syndrome, which is a monosomy situation, right? So that's an N. So mono is 1, right? Tri is 3. So this would be Turner syndrome is a monosomy situation. So for example, in a typical female, they have 2 X chromosomes. But with Turner syndrome, they only have 1 X chromosome. They don't have another X, but they also don't have a Y. So they have a monosomy. They have 1 instead of pairs. Where Down syndrome at pair 21, at the autosomal chromosome 21, instead of having a pair like normal, they have 3. They have the triplet there. Your patients that have these disorders, again, the Turner syndrome, they are usually sterile, okay, and have a variety of other side effects. So you can see on this graphic here, you don't need to memorize all of these or anything, but the one on the left here is Down syndrome, or the result of the trisomy 21 issue, so the triplet of chromosomes instead of a pair. You'll see the wide variety of symptoms that they have, from mental retardation, intestinal blockage, abnormal pelvis, decreased muscle tone, facial features are different. Okay? And the one on the right here is the Turner syndrome, so the monosomy, where there is only 1 X chromosome instead of 2. So again, a wide variety of phenotypic changes that causes. It causes physical differences, like there's particular facial features that are involved. They're shorter than normal. They can have some joint deformities. They don't menstruate because they're sterile. So this, just again, these what seem like maybe it shouldn't be that big of a deal can cause a wide variety of phenotypic changes. So these mutations can impact natural selection, right? If the phenotype that is affected, it's going to be subject to natural selection. So if it is beneficial for that particular environment, then it will be selected for. If it is not beneficial for that particular environment, it will be selected against. Okay? Prokaryotes, because prokaryotes don't officially do sexual reproduction, so they don't have crossing over to introduce variation. They are going to they're going to have a variety of ways where they can do some gene transfer that will help increase variation. That's not just solely based off of mutation. So we're going to look at these right here. So these are considered horizontal gene transfers. So going from prokaryote to prokaryote. So that's why it's a horizontal transfer. They're going from one organism to another organism, but it's the same organism. So the first thing that we have happening here is conjugation. Conjugation is this bottom picture right here. So we have a transfer of DNA due to cell contact. You can see these 2 bacterial cells here, and they're going to transfer some DNA throughout those plasmids. Remember, plasmids are those little circular pieces of DNA, and so they can transfer those plasmids from bacterial cell to bacterial cell. So that would increase some variation. Transformation, which is the top one here. So transformation is when they're going to pick up DNA from the environment. So DNA is in their environment. Maybe an organism has released some DNA. Okay, like for instance, you can see this one on the right has released the antibiotic resistant gene, and the one on the left is going to be picking up that DNA. So that would again cause some variation. We also have transduction. Transduction is the middle picture there. So with transduction, the viruses will help spread the DNA. So this virus here on the left, okay, has picked up some DNA from the bacterial cell, from the host cell. That virus then injects that DNA into the new host cell, and so that recipient cell will receive some of that DNA. And so again, we've got variation here. Also, within the organism, you can have transposition happening. So transposition, right, trans is across, right, so it's going to move positions. So the gene will actually move around on the chromosome. You can see that happening on the bottom here. So we have this gene that's going to move over to this other area of the chromosome. So it's not necessarily going to a whole different organism, but it's going to cause variety by its moving within the same organism. So viruses, and we've already said this should be redundant hopefully. So with that transduction, these viruses can combine and recombine genetic information when they affect the various host cells. And then for eukaryotes, organisms that reproduce sexually in particular, that is the benefit there is to increase genetic variation, and that allows, the more variation allows natural selection to come into play a little bit better. And so those things that are best suited, they're going to be evolutionary conserved. They're going to stay, okay? And they're going to be shared by many, many organisms. Let's look at an example here. So normally, scenarios where there is a detrimental effect for the homozygote, in particular the homozygous recessive, normally there won't be a lot of heterozygotes. To have a high frequency of heterozygotes is usually unusual for a particular genotype where it is a bad thing to be homozygous recessive. Because by having a lot of heterozygotes, we run the risk of increasing the frequency of this little a genotype. So that is an unusual thing to have a lot of heterozygotes when the homozygous recessive genotype is detrimental. An exception to that is going to be sickle cell anemia. So sickle cell anemia actually has a heterozygote advantage. So if you look at your maps on the bottom, you see your map of Africa there. And on the right, we have a map of where malaria is the most prevalent. And on the left, we have a map kind of tracking the sickle cell allele in populations in Africa. So in populations in Africa where I have malaria to be very common, homozygous dominant, okay, those people that are homozygous dominant, unfortunately, a lot of them are going to die of malaria. They're going to be infected by malaria, and they're going to die, so they're not going to reproduce, and so they're not going to pass their DNA on. Homozygous recessive genotypes, they are going to have a problem with actually having sickle cell anemia. So their malaria isn't necessarily the issue from them, but they're having all the side effects and the complications that go along with having full-blown sickle cell anemia. However, a heterozygote carrier is usually the most likely is going to be the one that to survive. Because a heterozygote carrier, yes, they will have some sickle cell issues because they will carry that sickle cell allele. However, that sickle cell allele offers protection from malaria. The way malaria works is the mosquito doesn't cause malaria. Malaria is caused by what's called the plasmodium, the parasite. And so the mosquito is the vector that transfers the parasite transfers the plasmodium from vector to vector, from organism to organism. So the plasmodium will then actually infects the blood cells. But when the blood cells are sickle-shaped, the plasmodium can't survive. So it is beneficial to have sickle-shaped cells in places where malaria is more common. However, you don't want full-blown sickle cell disease because now you have those problems. So in this particular instance, the heterozygote carrier is an advantage. It is a benefit. What about bacterial resistance or insecticide resistance? Right? So bacterial resistance is going to be referring specifically to bacteria that don't respond to antibiotics anymore. So your doctor prescribes a certain set of antibiotics. It doesn't kill the bacteria because they don't respond to that particular antibiotic anymore. These are caused by mutations in the bacterial DNA, and it's beneficial to the bacteria to have this mutation. So it's a positive mutation that will start to expand throughout the population because they're the ones that are going to be reproducing and surviving. So this will give our organisms with a mutation an advantage. And they're more likely to survive, they're more likely to reproduce. Therefore, their children will have that same resistance because they're going to have that same DNA. You see it with insecticides. You can see here their insecticide application. You spray the bug spray on the bugs, okay, but you happen to have this one that's able to survive. It happened to have a mutation, so it had a resistance to that particular insecticide. So that particular insect is able to survive. That one reproduces. Therefore, the surviving insects are going to have that DNA. They're going to have that resistance. So this is an instance here where these mutations are beneficial to these particular populations of organisms. So we'll continue practicing with mutations in class. Okay? And we will be tying it again to protein synthesis, and we'll practice some FRQs about this.